Very good morning to all. I wish you all a happy new year and you should get success in this new year. So starting with the DMA's first class of physiology. So we are going to enter into the basic classes. So this basic classes, why we have chosen is because in most of the exams, the questions is being targeted with the basics, whether it is a clinical or paraclinical, whatever it is, the basics is the must. So that is the reason why DMA has organized this free basic classes. So I hope this will be very much useful for all you guys. So in today's physiology class, we are going to cover few high yield chapters in the FMG or for physiology. Okay, so is it audible to everyone? Shall we start? Is my screen visible? If you have any queries, type in the chat box. Mute yourself, everyone. Okay, we will start. So starting with the high yield chapters in physiology. So these are all the important high yield chapters where you can see over here, the first concept is the general physiology and the nerve muscle physiology. So this general physiology, as you all know, this is the very important basic in physiology, which is accommodating 21.65% of the physiology chapters. So which is the most important in the physiology. And followed by that, you have the hematophysiology, respiratory system, endocrine system, cardiovascular system, and the other miscellaneous topics. So in the basic classes of this 6 days journey, so today's class we are going to cover the cell physiology and the introduction and important concepts in the nerve and muscle physiology, followed by endocrine physiology. Okay, so before moving into the cell physiology, we will see some important note on what is physiology. As you all always been studying, what is meant by physiology? It is the study about the body functions. Okay, so what is meant by physiology? We all know physiology is the study about the functions of our body parts. So in that, this physiological functions is the one which is playing a major role in maintaining the internal environment. So when you talk about the maintenance of internal environment, you will term that word as homeostasis. Okay. So this homeostasis, you also term it as milieu interior. So how this homeostasis is playing its role? This homeostasis, the maintenance of internal environment of our body, it is being maintained and mediated by the mechanism which you all called as feedback mechanism. So what is this feedback mechanism? Is whatever changes is happening in our body, it is getting the signal to the higher center. So this feedback mechanism, you have positive feedback mechanism and negative feedback mechanism. So we will be seeing the introduction of feedback mechanism. So what is meant by this feedback mechanism is, for example, in the maintenance of internal environment, in the homeostasis, you need few components. The first one is the receptor and the sensor, the controller, and the effector. So this receptor, this receptor is the one which is going to receive the signals from our body, from all the cells and the tissues, wherever it is. This receptor will sense the happenings in the regions. So these receptors will sense whether the functions of physiology is normal, whether the secretion of few hormones, chemicals, enzymes, neurotransmitters, whatever it is, and the secretions of the various organs like GIT, pancreas, and the kidney, all the functions is being received by the receptors which is present specifically in those target organs. So this receptor will receive the signal, and the receptor, once when it is receiving the signal, it will give the signal to the sensor, which is also called as afferent or sensory. So this receptor, whatever signal is being given from this receptor, the sensor will identify, and whether it is normal or if it is deviated like hypersecretion, hyposecretion, excess or deficiency, all those kind of signals is being sensed by these sensor, and the sensor will give the signal to the controller. As the name controller, this is the one which is going to analyze the signal which is given by this sensor. For example, if it is normal, it will maintain the function which is happening as such. If, for example, if there is any secretion of chemical which is excess, more than the required level, then the controller will try to reduce the specific chemical which is present in excess. And if it is deficient or if it is very low, then the controller will give the signal to the specific secretory region to produce more and more of those specific chemical, enzymes, hormones, or whatever it is. So this controller is the one which is analyzing the signal which is received by the receptor, and it will be sensed by the sensor, and then the controller will analyze. Once when it is analyzed, what the controller will do is the one which is now giving the signal. This is the motor signal or the efferent signal to the effector structure. This effector structure will now go and act on the target cell to correct or rectify the defect and bring it to the normal level. So this is how the homeostatic mechanism is functioning. So first and foremost, the receptor will sense the signal from the target cells and it will give it to the sensor. The sensor will sense the kind of signal which is received by the receptor and give it to the higher controlling center. This controller, the higher center, is the one which is having the ability to control the defect. And the controller, based upon the signal which is given by the sensor, it will give the signal to the effector to make the target cells rectify the defect. Finally, it is being brought back to the normal functioning. So this is called as feedback mechanisms. And moving on to the feedback mechanisms, you have positive feedback mechanism and the negative feedback mechanisms. So in the positive feedback mechanism and negative feedback mechanism, the most common feedback mechanism which is happening in our body is negative feedback mechanism. Most of the functions, it is maintained by this negative feedback mechanisms, whereas the positive feedback mechanism is very, very rare, and it has been happening only during some emergency conditions to maintain the homeostasis. And rest of the maintenance of the internal environment of the body is mediated by the negative feedback mechanisms. So this is the few basic introduction about the physiology and what is homeostasis and how it is maintained. Okay, so how the negative feedback mechanism is working and what is the very basic example of this negative feedback mechanism? We will see the thyroid level. For example, the secretion of thyroid hormones. So in the secretion of thyroid hormones, this is getting the signal always from the hypothalamus. And from the hypothalamus, the signal will reach the pituitary gland. And this pituitary gland will give the signal to the thyroid gland. So hypothalamus is giving the signal to the pituitary gland with the help of a hormone. You call it as thyrotrophin releasing hormone. And the anterior pituitary gland will get the signal from the thyrotrophin releasing hormone, and that will give signal to the thyroid gland to release T3 and T4. This T3 and T4, thyroxine and triiodothyronine, will reach the target cells. For example, if you encounter there is excess of T3 and T4, then immediately the feedback mechanism will start working. How it will work? Now it has analyzed, the receptor is receiving the signal that there is excess of T3 and T4. So immediately what will happen is, this excess T3 and T4 will give the signal to the hypothalamus, the negative feedback signal. Already I am in excess. I'm T3 and T4, I'm in excess. So you should stop giving the signal to the anterior pituitary. And this anterior pituitary, with the help of thyrotropin thyroid stimulating hormone TSH, it will also decrease, and then finally there will be decrease in the T3 and T4. So this is how the negative feedback mechanism is working its function. If in case, if T3, T4 is low, then the same mechanism will work, and instead of negative feedback signal, it will give positive signal to the hypothalamus and positive signal to the pituitary and positive signal by the TSH to encounter the defect, and it will increase the T3 and T4. So whether there is excessive secretion or if there is deficiency in the secretion, the low concentration, the hypersecretion or hyposecretion, whatever may be the defect, that will be received by the receptors on the target cells. Then through the feedback mechanism, the signal will reach the hypothalamus, pituitary, and finally the thyroid gland to rectify the defect. So this is how the negative feedback mechanism is happening. So this is one of the example for negative feedback mechanism. As like this, all the negative feedback mechanism is happening in our body to maintain the internal environment. And now moving on to the positive feedback mechanism. So in the positive feedback mechanism, we will see the production of oxytocin. So this is the hormone of the posterior pituitary gland. Okay, so this hormone, when it will be produced, is especially at the time of labor. So at the time of labor, when there is a contraction of uterine muscles, so the stretch of the uterine muscles, it is sensed by the receptor, and the receptor, now it is giving signal to the sensor and then to the controller. In case of oxytocin, the controller here is the hypothalamus and posterior pituitary gland. So the stretch of the uterine muscles during the contraction of the delivery. So the stretch of the muscles is received by the stretch receptors and it is giving the signal to the sensor. The sensor will now give it to the higher center, the hypothalamus. The hypothalamus followed by the posterior pituitary gland. Hypothalamus is the region where there is synthesis of oxytocin and it is stored in the posterior pituitary gland, and the hypothalamus will give the signal to the posterior pituitary gland to release more and more of oxytocin. So this oxytocin will go and further reach the uterine muscles and favor the process of labor. And once when the baby is delivered, after the delivery, so all over here you are getting the positive signal. So that is helping in the labor. After the delivery, immediately the contraction signal, the stretching of the uterine muscle is lost. So it will start decreasing the production of oxytocin. And this kind of example is for the positive feedback mechanism. I hope everyone is clear with this feedback mechanisms. Yes, everyone is clear. The very basic introduction of physiology, homeostasis we have covered. And now we will move on to the cell physiology. So in the cell physiology, as you all always studying the definition of a cell. What is a cell? It is the structural and the functional unit. Whether it is any kind of cell, whether it is a nerve cell, muscle cell, or any other type of cell, secretory cells, whatever it is, that is the structural and functional unit of the specific target structure. Okay. So before going into the systems of the physiology, we have all the systems. We should know the basic of all those systems. The first and foremost is the cell, and all the group of cells will join together and form the tissue, like nervous tissue, muscle tissue. And then the multiples of tissues which is having the similar functions will join and it forms the organs. Example like stomach, liver, pancreas, and all those organs which is having the similar functions. For example, the organs of the digestive system. It starts from the mouth, esophagus, stomach, then the duodenum, small intestine, large intestine, liver, pancreas. All these are different type of organs, but they are all involved in the digestive functions. So all those group of organs which is having the similar functions, we will call it as a system. Like digestive system, when you talk about the renal system, it starts with the kidney, ureter, urinary bladder, urethra. All those are the different organs which is performing the excretory function or the renal function in the process of urine formation and excretion. So that you call it as a renal system. So cell is the basic structural and functional unit of the living system. So all the cells which is having the unique functions will form the tissue, and the tissues which is having the similar function will form the organs, and the organs which is having the similar functions will form the systems. So we have so many systems. Starts with the blood, then the GIT, gastrointestinal system, cardiovascular system, renal system, endocrine system, nervous system. So this system is being formed by the cells, the tissues and the organs having the similar functions. This is a very basic, but still I want to insist on this. So now, what is the cell? Which is the structural and functional unit of cell. Before going into the cell and its organelles and the functions of all those organelles, we should know about the cell membrane. So talking about the cell membrane, so this cell membrane, it is made up of three important contents. So cell membrane, it is made up of three important contents. What are all those? When you talk about the cell membrane, you have the first and foremost lipids and proteins, followed by carbohydrates. Okay, so lipids, proteins, and carbohydrates are the three major contents of the cell membrane. We will go on to the diagrammatic representation in a couple of minutes. So first we will see what is a cell membrane made up of. This cell membrane is made up of lipids, proteins, and the carbohydrates. So this cell membrane, this lipids, it is constituting nearly 42% of the cell membrane, and the proteins, which is constituting 55% of the cell membrane, and the carbohydrate, which constitutes only 3%. Okay, so this is what is the components of the cell membrane. So this cell membrane, now we will move on to the details of lipids first. So this lipids, it is made up of phospholipids, cholesterol, they are all the different type of lipids, and other trace amount of other forms of lipids. So among this, phospholipids play a major role in the formation of the cell membrane. So all those yellow colored structures in this picture, what you see, they are all the phospholipids. So when you see all over these, they are the phospholipids. And then all those structures which is sticking here and there, they are the cholesterol and other structures we will see one by one. So this phospholipids, when you take an individual structure of the phospholipid, you will have a polar and the non-polar structure. So this is the structure of a single unit of a phospholipid. So in the single unit of the phospholipid, this you call as head and the tail portion. So this head portion is polar. In the sense, it is hydrophilic, which has affinity with the water. Whereas the tail portion, it is non-polar, where it is hydrophobic, which is having fear with the water. When you look into the structures, see, you have one phospholipid arranged in this manner and the another phospholipid which is arranged in the inverse manner. So the tail portion of both the phospholipids is facing inside. The tail part is facing inside. That is the non-polar. Whereas the head portion of both the phospholipids are facing outside. So why it is arranged so is because the head portion is polar. On either side of the cell membrane, you have the fluids. The one is extracellular fluid and the other one you have intracellular fluid. So that is the reason this tail portion, which is non-polar, is always arranged in the inside of the cell membrane. It does not need the fluids. It is having fear with the water. So it don't want to face the fluid, whether it is intracellular or extracellular. So that is the reason why the phospholipids are arranged in this inverse manner, one in the straight manner and the other in the inverse manner. It is because the tail portion of the phospholipid is non-polar, which is hydrophobic. Okay, and this is the one which is giving you the structure of the phospholipid as the structure of the cell membrane. You call it as fluid mosaic model. Okay, and you also call this sandwich model of cell membrane. This is because of the arrangement of the phospholipids, which is always making the tail portion of the phospholipids to remain away from the fluid, either it is intracellular fluid or the extracellular fluid. Okay, so that is the content about the lipids. And now moving on to the proteins. So in the proteins also you have two types of proteins. One is the integral proteins and the other one is the peripheral proteins. As the name integral proteins, these integral proteins, it covers the entire thickness of the cell membrane, which is present from ECF up to the ICF. So this is another example of the integral proteins. So this integral proteins, it covers the entire thickness of the cell membrane. So it also acts as a transmembrane. Okay, so these integral proteins, since it covers the entire thickness of the cell membrane, it acts as a ionic channels, or it acts as a carrier proteins. It also acts as a transport pumps. So this integral protein, it is present all over the thickness of the cell membrane from ECF up to the ICF. So it acts as a transmembrane protein. So since it is having the connection with both ECF and ICF, it makes the molecules to move from inside to outside or from outside to inside. It makes the movement of the molecules, solvents and all the kind of transport materials from ECF to ICF or from ICF to ECF. Whereas the peripheral proteins, as the name peripheral, it is not present towards the entire thickness of the cell membrane. It is located peripherally. Okay, so this acts as cell adhesion molecules. And it also acts as a anchoring proteins between the cells. Okay, and this also acts as antigens. Some of the cells will expose its antigen. For example, the RBC cells, red blood cells, which have its antigen structure in the cell membrane, which helps us to identify the blood typing. So these peripheral proteins, they do not have the structure which is arranged with the entire thickness of the cell membrane, but they are arranged only peripherally. So since they are arranged peripherally, they act as cell adhesion molecules and it acts as an anchoring. It helps in the anchoring of one cell with the another cell, and it also acts as a surface antigens. Okay, and some of the peripheral proteins will also act as a surface receptors. Okay, so this is what is about the proteins. And moving on to the carbohydrates. This carbohydrates, it constitutes only 3% of the cell membrane, and it helps in anchors between the extracellular matrix and adjacent cell. So when you talk about the carbohydrates, these are all the carbohydrate molecules. Okay, so the carbohydrates not only attach it to the lipids, they are also attached to the proteins. So you call it as glycolipids or glycoproteins. So the carbohydrate which is attached to the lipids, you term it as glycolipids, and those which are attached to the proteins, you term it as a glycoproteins. So overall, these carbohydrates, it forms the glycocalyx. Okay, so this carbohydrate, it covers the entire unit of the cell membrane. So because of that, it covers like wrapping of all the fluids, the phospholipids, they are the lipids. So it will not have the stability. So it will be flip-flop mechanism, and all the cholesterol and other form of lipids, everything is fatty substances. So it will not give the stable structure to the cell membrane. And same way, the proteins are not present towards the entire of the cell membrane. You have the integral proteins, peripheral proteins here and there. So this will also not give the stability to the structure of the cell membrane. Whereas when the glycocalyx is being formed, which wraps the entire structure of the cell membrane, and it maintains the stability. It gives the stability to the cell, and it maintains the shape, structure, and the shape of the cell. So when you talk about the cell, you can determine this is a cell. This stable structure, the stability to the cell membrane is given by the carbohydrates. Okay, so this is what is the introduction about the cell membrane. So when you talk about the composition of the contents of the cell membrane, the lipid constitutes 42%, whereas the protein constitutes 55%. Okay, so this is common in most of the cells, but there is one small exception in case of myelins. Myelin sheath, it is the covering, insulating structure of the axons of the nerve fibers. So these myelins, in this case, the proteins is less than that of the lipids. Whereas in all the other cases, the lipids is playing a major. In all the other cases, protein plays a 55%, except in the myelins, the protein constitutes more, protein is less and the lipids is high. This high lipid content helps in the insulating of the signals. It favors good insulation. Okay, that is the exception of the concentration of the lipids and protein contents in the cell membrane. Okay, clear with this? Yes. So now we will move on to the extracellular matrix. So there is a small change over here. This is not the fluid. This is extracellular matrix. So extracellular matrix is the part which is present outside the cell. So this extracellular matrix, when you talk about the extracellular matrix, they are the structures which is present outside the cell. For example, this is a cell, and this is another cell, and this is another cell. And those structures which is present in between all the cells, so these forms the extracellular matrix. So this extracellular matrix will also have few proteins and the structures which makes the cell, one cell to adhere with the another cell. So this is the one which is required for making all the cells to join together and form the tissues. So what is this extracellular matrix made up of? So this extracellular matrix, that is the space between the cells. Okay, so it is also known as connective tissue. So the space between the cells, which you call it as extracellular matrix, it is also called as a connective tissue. So what are all the functions of this extracellular matrix? So what function is being mediated by this extracellular matrix is so the first one, it gives the structural support and it acts as a communication space between the cells, and it also helps in the transport of substances either between ECF to the ICF or between the cells. Okay, so you have to transport few substances between the cells or between the cell intracellular fluid and the extracellular fluid. It depends upon the function whatever it is required. So all those is being functioned by this extracellular matrix. So what is this extracellular matrix made up of? This extracellular matrix, it is made up of structural proteins, which is like collagen, okay, elastin and fibrillin. So collagen, elastin, fibrillin, it forms the structural proteins of the extracellular matrix, which gives the structural support. And it also have the space between the cells. So it means it acts as a connective tissue to adhere one cell with the another cell. So that is given by the adhesive proteins. So these adhesive glycoprotein, it is formed by fibronectin. At last, you have glycans, which also helps in the structural support, especially the hyaluronic acid and sulfatase. Okay, so this fibrillin, among this all contents of the extracellular matrix, the fibrillin is having a clinical significance where when there is a mutation of this fibrillin protein that results in a clinical syndrome, you call it as Marfan syndrome. Remember, when there is a mutation of this fibrillin protein, which is the structural protein of the extracellular matrix, so that results in the clinical condition called as Marfan syndrome. So Marfan syndrome, what are all the clinical features you will encounter in case of Marfan syndrome is the person will have long limbs, crowded teeth, and very gigantic nature, which you call as Marfanoid habitus. So this is one of the clinical scenario which you will encounter when there is a mutation of the fibrillin protein of the extracellular matrix. Okay, so with this, the extracellular matrix and its important concepts we have seen. So main thing is it maintains the structural support, it favors the connection between the cells and favors the transport. So you have structural proteins, adhesive proteins and the glycans. Among all these things, the structural protein, collagen, elastin, fibrillin, when there is a mutation of this fibrillin protein, when there is a mutation, it results in the clinical feature. You call it as a Marfan syndrome. So that is the peculiar thing you have to remember with this extracellular matrix. Okay, and now moving on to the cell organelles. So in the cell organelles, I hope everyone can label the parts of the cell organelles. So first we will label, this is the nucleus, and in this last FMGE exam December 2021. There is a question with this cell organelles. I will tell you that now. So you should know the cell organelles and its basic functions. Okay. And inside the nucleus you have nucleolus, and these are Golgi apparatus, the mitochondria, and this is rough endoplasmic reticulum because you have the ribosomes attached to this endoplasmic reticulum, and these are lysosomes, secretory granules which contains the enzymes and other microfilaments, and the microtubules. Okay, so some basic important cellular organelles we have marked over here, and we will be seeing few important cellular organelles, its functions. Okay, in the past FMGE exam, there is a question which is considering with the lysosomes. These lysosomes are the structures which contains the digestive enzymes. So these digestive enzymes, these lysosomes will have its role in the immunity also. That is the question which was being asked in this 2021 December FMGE exam. Which of the cellular organelle is having its role in the immune mechanism? So the answer is lysosome. So why this lysosome is considered to be the one having role in the immunity is because this is performing its function with the phagocytosis. Okay, so this phagocytosis, it is mediated by the white blood cells, the neutrophils and the macrophages, monocytes, to engulf the foreign material, to engulf the bacteria, viruses or any other foreign material. It will make the phagolysosome with the phagocytic cell, and it will release all the digestive enzymes and finally kill the foreign material. So since it is having its role in fighting against the foreign substances, this is the one which is having its role in the immunity. So this is the very basic which we are all studying from your higher secondary onwards. The lysosomes, phagocytic cell fusing with the lysosome, forming the phagolysosome, and then it will release all the digestive enzymes, kill the cell debris, and then it will encounter the foreign material. And apart from this immune function, defense mechanism, the lysosomes is also having its role in the physiological mechanisms to make the digestion of the cell debris. For example, RBC, the normal lifespan is 120 days. After the 120 days, you have to digest all those cell debris, the remnants of the RBCs, and then you have to encounter. So this lysosome is not only having its role in the immune mechanism, it is also required during the maintenance of the physiological environment. So this is how you will get the basic questions in the physiology. Okay, so now we will move on to the few cellular organelles, and the most important thing which we are all going to see now in today's class is the rough endoplasmic reticulum. So rough endoplasmic reticulum, you have the ribosomes in its structures. So these ribosomes are the one which is involved in the protein synthesis. So what are all the proteins or the chemicals which is synthesized by these ribosomes is like insulin and some hormones. And it also produces the lysosomal enzymes and also in the collagen synthesis. So these are all the some examples of those substances which is produced by the rough endoplasmic reticulum. And these rough endoplasmic reticulum, since it is having its role in all these digestive enzymes and all those things, where you, in which cell, which organ, you will have more and more of rough endoplasmic reticulum. It is mainly in the goblet cells of the small intestine and also in the plasma cells. So which has rich composition of the rough endoplasmic reticulum. So in case of neurons, where you will see this rough endoplasmic reticulum, it is present as nissl bodies. So which has the rich source of the enzymes. Okay. So this is about the rough endoplasmic reticulum. And now moving on to the smooth endoplasmic reticulum. I will make a note over here of smooth endoplasmic reticulum. So smooth endoplasmic reticulum, as the name, it is smooth, there is no ribosomes. So this is also involved in the synthesis of few hormones, especially the steroid hormones. And it is also having its major role in the detoxification of drugs. Okay. So since it is involved in the detoxification of drugs, which is the rich source of smooth endoplasmic reticulum? It is the liver. Okay. So here it is having major role in hormones and enzymes. This is rich in the goblet cells and also in the plasma cells. Whereas smooth endoplasmic reticulum, it is having its major role in the detoxification function. Detoxification is mediated by the organ which you call as a liver. So this liver hepatocytes contains rich source of smooth endoplasmic reticulum. And since it is involved in the synthesis of steroid hormones, it is also rich in adrenal glands, especially the adrenal cortex, which is having its role in the synthesis of glucocorticoids, which are the steroid hormones. So liver and the adrenal gland has rich source of smooth endoplasmic reticulum. And now coming on to the Golgi apparatus. So whatever proteins, enzymes, hormones, chemicals you are all synthesizing from the smooth and the rough endoplasmic reticulum, you should pack all those materials. So that is being mediated by this Golgi apparatus. Okay. So this will pack all the enzymes, chemicals, proteins which is synthesized by the rough and the smooth endoplasmic reticulum and make it within the cell in the form of secretory granules, lysosomal enzymes, and then it will give it to the target cell when there is a need for all those specific chemicals. So that packing of the substances synthesized within the endoplasmic reticulum is mediated by this Golgi apparatus. So apart from this, all the other basics of cell granules we will see detailed in the regular classes. So this is about the cell and its cellular organelles. And coming to the peroxisomes, these peroxisomes is different from the lysosomal enzymes. Lysosomal enzymes are the hydrolase enzymes. When you see the lysosomal structures, they have the chemicals, they are hydrolase enzymes. Whereas these peroxisomes have oxidative enzymes. Okay, which prevents from the oxidation process. So it is helpful, it acts as an antioxidant structure. So all the oxidative chemicals which is produced within our body, it is being removed by these peroxisomal enzymes. So these peroxisomals also called as antioxidants. It contains antioxidants which removes our body from the threat of oxidative damage. Okay, so that is being mainly mediated especially by the oxidative phosphorylation of very long chain fatty acids, branched chain fatty acids, and some amino acids, alcohols. So they are all involved in producing this oxidative enzymes. And the most common one is hydrogen peroxide, which also acts as an antimicrobial agent and also as an oxidizing agent, which removes our body from the oxidative stress and the oxidative damage from the oxidants. So these peroxisomal enzymes has its major role as antioxidants, and it prevents the body from the oxidative damage. Okay, so this oxidative damage is very common nowadays because of the excess of stress and the nature of work, all those things. So up to this, the cell membrane and the cellular organelles, we are finishing. And now we will move on to the cell junctions. So cell junctions also, we have so many types. The common is the tight junctions, adherent junctions, desmosomes, hemidesmosomes, and the gap junctions. The peculiar functions of all these things we will see now. So coming to the tight junctions, they are also called as zonula occludens. Okay, so this, as the name tight junctions, there is no passage of molecules. So it is the junction between the one membrane of the cell to the another membrane, but it will not allow the movement of any molecules. It will not allow the passage of any molecules through the tight junctions. Okay, and next one is the adherents. Adherents, they are also called as zonula adherents, which is the intermediate junction. Okay, this tight junction is present at the top of the cell, and then below the tight junction you will have the zonula adherents. They are the intermediate junctions, and next, followed by the intermediate junctions, you have the desmosomes, which you call as, also called as macula adherents. Okay, so this macula adherents, it functions as the junction between the cells. Okay, and another important thing is this macula adherents produces a protein called as desmoglein. Okay, so this desmoglein is present in the desmosomes. When there is a mutation or any other defect of this protein, it results in the clinical feature as rashes or erosions, which you call as pemphigus vulgaris. So this is pemphigus vulgaris, because it has produced all the erosions and the rashes all over the skin. So this is because of the mutation of desmoglein protein. Okay, so that is one of the important feature you have to remember in case of this desmoglein. And followed by desmoglein, you have the hemidesmosomes. Okay, so this hemidesmosomes, as you can see in this picture, so this is a cell and it is attaching with the cellular matrix. So this hemidesmosomes, it is the junction between the cell and the matrix, extracellular matrix. Okay, so this hemidesmosomes also has its own protein, but in case of any mutation or any other abnormality, there will be production of anti-hemidesmosomal antibodies. So this is the junction between the cell and the extracellular matrix, where there is a fluid in between. So because of that, when there is a formation of this anti-hemidesmosomal antibodies, it results in the condition of the blisters, fluid blisters, because it is the junction between the extracellular matrix and the cell where you will find the fluids in between both the junctions. So that is the reason why you will encounter the fluid blisters in case of hemidesmosomal defect, and that you call it as a bullous pemphigoid. Remember, pemphigus vulgaris is rashes and the erosions because it is between the cells. There is no fluid in between these two junctions. Whereas in case of the hemidesmosomes, it is the structure which is present between the cell and the extracellular matrix. And that is the reason why you will encounter the fluid blisters as bullous pemphigoid. So this is the bullous pemphigoid where you can encounter the fluid blisters. Whereas in case of this pemphigus vulgaris, erosions and the rashes alone is being seen. But here you will encounter all the fluid blisters. So this is how you have to identify what are all the things happening. And coming on to the another important cell junction, that is called the gap junctions. As the name gap junctions, it allows the movement of molecules between the junctions. Okay, so that is the junctions which contains the major protein called as connexins. Okay, so these connexins and the gap junctions, it is rich in the cardiac myocytes. Since this cardiac myocytes is rich in the gap junctions, it makes the cardiac muscle to function in a unique manner. When the cardiac muscle is contracting, relaxing, it contracts on the whole. It acts as a syncytial fiber. It will not function as individual with the individual cardiac muscle. So cardiac muscle is having its syncytial fibers because this cardiac muscle is having rich source of the gap junctions, and that is favored by the help of protein called as connexins, which helps in the fast impulse conduction. Okay, so when there is any defect in this connexin protein, you will encounter the condition called as Charcot-Marie tooth disease. That is because of the mutation in the connexin protein. So cell junctions, we have seen desmoglin in the desmosomes. When there is defect, encountering of pemphigus vulgaris, and in case of hemidesmosomes, you will encounter bullous pemphigoid, whereas in case of gap junctions, you will encounter Charcot-Marie tooth diseases. Okay, so up to this, the cell junctions is over. And coming to the fluid physiology, we all know the cell, and inside the cell and outside the cell. Okay, inside the cell you have intracellular fluid and outside you have extracellular fluid. Whereas in between the cells you have the fluid, you call it as interstitial fluid. So all these is now going to give you the fluid physiology. You can see this picture. So this is the cell, where the cell and the fluid inside is the intracellular fluid, whereas in between the cells you have the interstitial fluid. Along with that, you have the intravascular fluid called as plasma. and the interstitial fluid together with the plasma, you call it as extracellular fluid. And this fluid composition, you have to remember important content in this is the total body fluid. You have 60%. So among the total body weight, the 60% of the weight is being given by the fluids. So in that, intracellular fluid and extracellular fluid, intracellular fluid constitutes 40%, whereas the extracellular fluid constitutes 20%. So this composition you will remember as 60, 40, 20 rule. So in order to make it remember easily which is 60, which is 40 and which is 20. This is total body fluid, intracellular fluid, extracellular fluid. You can remember with the mnemonic type. Okay, 60, 40 and 20 rule. So this is very, very important. So this composition 60, 40, 20 was being asked in one of the FMG question. So which is 40, which is 20, which is 60. Don't get confusion. Remember with the mnemonic type. Total body fluid 60, I is 40 intracellular fluid, E is extracellular fluid. And this extracellular fluid includes both interstitial fluid and the intravascular fluid, that is the plasma. Okay, so this is the important thing. And then coming to the some important contents which is present within the intracellular fluid and the extracellular fluid. So the most important is the sodium, which should always be within 135 to 145 milliequivalents per liter. It is more in the extracellular fluid. Okay, and the potassium, which always be in excess in the intracellular fluid, and the chloride, that is 98 to 100 milliequivalents per liter. Clinically, we will always see this three contents: sodium, potassium and chloride. If there is any defect in the sodium, potassium and chloride composition, that should be immediately rectified, because they are all the one electrolytes which is having its major role in maintenance of most of the functions in our body. Okay, so this is the introduction part of the fluid physiology. And moving on to the cell transport. We have seen cell junctions, extracellular matrix and the cell membrane, channel proteins and the integral proteins, all those things which favors the movement of molecules, movement of substances between the cell, in between the cell and the extracellular fluid, in between the extracellular fluid and the intracellular fluid. So all those transport mechanisms we have seen. So now we are going to see how this transport mechanisms is taking place and what are all those substances or the chemicals which is being transported between the cells, inside to the outside the cell, all those things. Okay, so first and foremost, this cell transport, it is of passive transport and the active transport. So passive transport and active transport. So what is meant by passive transport and the active transport? As the name passive, it will pass without any energy. It passes freely and there is no need for the energy. So you also term it as downhill movement. Whereas active transport, active means it requires energy. It will not move freely because it needs the channels or the pumps. Okay, and since it requires energy, it is called as uphill movement. The very classic example, a person is climbing the mountain. Okay, in a bicycle? The person requires energy. Energy is required for the person to ride the bicycle to go up to towards the mountain from the base. But at the same time, if the person is riding the bicycle from the top of the mountain towards the base, there is no need for energy. He can just sit happily and just hold the cycle enough. So that is called as downhill movement. That is the passive movement. But here, uphill movement, active movement requires energy. So this is the classic example which is being given for the active transport and the passive transport. Okay, so now you know what is active transport and what is passive transport. So first we will see the types which is present in the passive transport. That is simple diffusion, osmosis and facilitated diffusion. In simple diffusion, as the name, it will just diffuse through the cell membrane. So when it is diffusing through the cell membrane, we have seen in the cell membrane there are lipids, proteins, which is having its major composition. So lipids will freely allow the lipid soluble substances like oxygen and carbon dioxide. For the transport of oxygen and carbon dioxide between the from one side of the cell membrane to the other side of the cell membrane, you don't need any energy. You don't need any expenditure of pumps or the channels, because oxygen and carbon dioxide are the very important gas exchange which is required in our body for the maintenance of the functions. So they are all the lipid soluble substances. It will diffuse freely through the lipid bilayer, the phospholipid layer of the cell membrane. That is simple diffusion. And in case of proteins, the protein substances like amino acids and few glucose molecules and the electrolytes, they are all the protein substances, and the water molecules. Okay, so all these are the protein molecules which can diffuse through the protein layer, because the integral proteins which we have seen, it is present towards the entire thickness of the cell membrane. So these integral proteins is also acts as a channels, and it allows the movement of protein molecules from one side of the cell membrane to the other side of the cell membrane. And the another important thing is the facilitated diffusion. So when you talk about the simple diffusion, the cells is allowing the passage of the molecules passively to diffuse. But there is few limitation for the simple diffusion. What is that limitation is, when you have the lipid bilayer, the phospholipid bilayer, you have like this. So when you have, if you have the protein lipid molecules, for example, some lipid molecule is present over here. It has to pass by the simple diffusion. It will pass only when it is of smaller than the pore size of this lipid layer. If the lipid molecule is bigger in size, it cannot travel through this space between the phospholipids. So such kind of lipid molecules or the protein molecules, which are all the macromolecules, which has the size bigger than that of the pores which is present in the lipid bilayer or the protein layer, that can be transported by the help of facilitated diffusion. This facilitated diffusion also happens passively with the help of some carrier molecules. Okay, so this carrier molecule is required in case of facilitated diffusion of passive transport. Okay, so that is what is the passive transport mechanism. And now moving on to the active transport mechanism. So in active transport mechanism, also you have the transport mechanism with the help of the pumps and through the vesicles. Vesicles in the sense, these vesicles is going to have some chemicals or enzymes or hormones or any neurotransmitters which is present inside the cell and through the channels or the pumps. So this active transport basically have primary active transport and the secondary active transport. So both active transport mechanism, primary or secondary, requires energy. But in case of primary, the most of energy is expenditured in the form of ATP. Whereas in case of secondary, also it is ATP, but it is dependent on the ionic gradient. This also requires ATP, but this is specifically dependent on the ionic gradient, the concentration gradient of the ionic molecules, which is about to be transported between the cell membrane. Okay, so that is active transport. And apart from these two differences of energy molecule in primary and secondary active transport, this secondary active transport is further divided into two subtypes. One is symport and antiport. Symport you also called as co-transport, and this is counter transport. Symport and antiport. So I will tell you the example for this symport and the antiport. I'm sorry for this. When I zoom out, this picture is not clear. So I will draw and explain you. So when you take the cell or the cell membrane, so when you take the cell membrane, okay, so if you want to transport the substances from one side of the cell to the other side, you consider this as ECF and this as ICF. And in case of active transport mechanism, you want energy in the form of ATP. Okay. So if you want to transport the substances from ECF to ICF, for example, sodium along with this glucose. Okay. Always in case of active transport mechanism, most of the transport will not happen in one way with the one molecule. Okay. Either it will carry another molecule in the same side, or it will be like one molecule is coming inside and the other molecule will go outside. Example with the sodium, potassium. This also requires ATP. Okay. So all the case of transport, maximum level, it requires sodium. It will be like sodium glucose transporter, sodium amino acid transport, sodium water transport, sodium potassium, sodium H+. So most of the transport pumps or the ionic channels is dependent with the sodium. That is the reason why in case of the fluid physiology, we have seen sodium is playing a major role in the extracellular fluid. This sodium is required for the basic functioning of the cell. So if a person is being admitted in the hospital, you have to encounter the sodium concentration and immediately you have to rectify the sodium defect if the person is having. It should not be higher, it should not be lower. Okay. And that is another reason if a person is having dehydration, the sodium will move along with the water, because in case of dehydration, whether it is of vomiting or diarrhea, excess, whatever it is, there will be loss of water. Along this water will not move alone. It will always carry sodium along with that. So when there is a water loss, when you encounter dehydration, it means that the person's electrolytes is also less. So before identifying the cause of the dehydration, and finding all those blood parameters and analyzing all the cause, what is the cause for the dehydration. Before doing all these things, immediately you have to rehydrate by giving the electrolytes. Okay, if you give the electrolytes, it contains sodium, potassium, magnesium, chloride. So if it is being maintained, then the water level is also maintained and dehydration will be rectified. This is the first and foremost thing which you have to do. Immediately after that, you can do all the other step-by-step events to find the cause of the dehydration and rectify it. Okay, so when coming to the active and primary and secondary active transport, if the molecule is moving in the same direction, that is called as symport. And see over here, the sodium is moving from ECF to ICF, whereas the potassium is moving from ICF to ECF. So this also with the two molecules, but in different directions, that you call as antipode. This is co-transport and this is counter-transport. So that is the two different types of transport in the secondary active mechanisms. So these primary mechanism and the secondary mechanism of active transport is dealing with the solid molecules. When you deal with the vesicles, okay, so this is going to deal mainly with the fluids. The fluids, how it has been transported. If it is a solid molecule, electrolytes, potassium, magnesium, sodium, glucose, amino acids, they are all the solid substances. So you require channels and the pumps for transporting all those things. If you want to transport fluids, which is present inside the vesicles, you need the special carrier transport that is called as exocytosis and the endocytosis, which you call as vesicular transport. So exocytosis and endocytosis, vesicular transport. Exocytosis is the process of, this is the example of exocytosis. And this is endocytosis. And I have another one more picture for vesicular transport here. Okay, so exocytosis. So here what happens? Exocytosis mainly for the bacteria, or in between the synaptic membrane, you have to transport the synaptic vesicles and the synaptic neurotransmitters. So all those is being transported by the exocytosis mechanism. Okay, first it will be encountered. It is not only for the bacteria, remember that. Then it forms the phagosome, fusing with the lysosome and forming the phagolysosome. This phagolysosome will come and rupture with the other end of the cell membrane, and finally the molecules will be released. It was like the cell is vomiting. So that is called as exocytosis. And pinocytosis, it is like cell drinking. Okay, in case of pinocytosis, where you will encounter this exocytosis, mainly you will encounter with the macrophage cells, phagocytes, and that is mainly mediated by most of the white blood cells. Whereas in case of pinocytosis, cell drinking, it means the cell is absorbing some of the fluid molecules, absorbing some of the molecules. So that absorption process mainly takes place in the small intestine, microvilli, and also in the kidney ductal cells to reabsorb, tubular reabsorption, and also in the absorption of fat droplets. So these are few examples where the cell drinking is taking place. See over here in the extracellular fluid, you have the molecules to be reabsorbed. So it will go into the cell membrane, form the pinocytic vesicle, and it will remain as a cellular organelle. So in order to mediate this pinocytosis, for those molecules to come and bind with the cell membrane, you need specific proteins which is called as, which acts as a receptor. They are clathrin proteins and caveolin proteins. So those endocytosis process which is mediated by these proteins, you also called as receptor mediated endocytosis. Okay, so that is endocytosis mediated by the clathrin and caveolin. They form the receptors over here. So those receptors will favor the molecules to come and bind all over these structures. Okay, so that is vesicular transport. And one more thing we have missed out over here is the osmosis in the passive transport. So osmosis, what is osmosis? It is one of the passive transport mechanism. So first of all, when you talk about the definition of osmosis, it is the diffusion of water or solvents through the semi-permeable membrane. Okay, towards the higher concentration. That is osmosis. Either diffusion of water or the solvents through the semi-permeable membrane towards the higher concentration is called as osmosis. This movement also does not require any energy. Okay, so that is called as osmosis. And see over here, when you put a cell, this is the example for the osmosis. When you put a cell inside the fluid, so the solution may be either of hypertonic, isotonic or hypotonic. What is meant by this tonicity is when the concentration of the solution is equal to that of plasma, that is isotonic. When the concentration of solvents which is inside the solution is less than that of the plasma concentration, that is hypotonic. If it is higher, you term it as hypertonic solution. Okay, when the high tonicity of the solution is based upon the amount of solutes compared with to that of the plasma. Okay, so normally the plasma this will maintain the osmolality, and the normal plasma osmolality is approximately 290 milliosmoles per kg. So this 290 milliosmoles per kg, this value is very important when you deal with the renal physiology in the urine concentration and dilution. When you are encountering the renal physiology, this plasmo-osmolality plays a major role to form the concentrated or diluted urine. So what is this now going to happen if you put the cell in the isotonic solution? The movement will remain same. The cell will maintain in its normal nature. Instead, if you put it in a hypotonic solution, all the water molecules will move outside because of the movement towards the higher concentration. This is the movement towards the higher concentration. So the fluids which is present inside, the water which is present inside the cell is now going outside. So it causes shrinkage of the cell. And at the other end, if you put the cell in the hypertonic solution, the movement of the fluids is towards the cell. So it will cause enlargement of the cell at one stage, lysis of the cell will take place. So with this, the cell transport is over. Okay, clear with this? And now the cell physiology is over and we will move on to the nerve and muscle physiology. Up to this, clear everyone? Yes? Everybody is awake? Yes? Okay. It's good that I'm getting the reply. Okay. So now we will continue with the nerve physiology. I will try to finish in half an hour and I will give you just 2 to 3 minutes break. Then we will start with the endocrine system. So moving on to the nerve and muscle physiology. So before going into this ionics and the flow of current, all those things, we will see the structure of a neuron. When you look into the structure of a neuron, you have the cell body and the dendrites. Followed, these dendrites are the short processes. Okay, the extensions from the cell body you call as processes. In a neuron, you have short process and the long processes. The short processes you call as the dendrites, and the cell body you also call as soma, and the long process you call as axons. At the end of long process, you have the terminals of the axon that you call as synaptic knobs. Okay, so when you look into the axon, you have the myelin sheath over here. So this is the axon. So this axon is covered by the myelin sheath. So this myelin sheath, it functions as a protective sheath for the axon, and also it functions for fast transmission of signal from the cell body to the adjacent cell. So the faster transmission of the signal is mediated by this myelin sheath. So this myelin sheath is made up of schwann cells in case of peripheral nervous system, whereas the myelination is formed by the oligodendrocytes, which is one of the glial cell in case of central nervous system. Okay, in case of peripheral nervous system, one myelin sheath is formed by one schwann cells, meaning that a schwann cell will form myelination of one neuron alone in case of peripheral nervous system. Whereas in case of central nervous system, one oligodendrocytes will form myelination for multiple neurons. Okay, in the regular physiology classes, when we are starting with the nervous system, we will tell you the details of all the glial cells and the oligodendrocytes, how it is forming the myelination and all those things. Okay, so here we will just see up to this, and this is the basic structure of a neuron. So now we are going to see, it is the place where the signal transmission is taking place. The signal transmission is going to take place with the help of the myelin sheath. And how it is going to take place? In a nerve fiber, you have the myelin sheath. Okay, the signal is being produced at the axon hillock. That is, the axon hillock is the place where the axon starts from the nerve fiber. That is the place where there is a initiation of the impulse. This impulse will move the space between one myelin sheath and the other myelin sheath. You don't have the myelin sheath in between the spaces. So those spaces is called as node of Ranvier. So that is the place where the jumping of potential will take place. So this favors faster conduction of impulse. Only in the place where you have the myelinated neuron. If it is non-myelinated neuron, the impulse transmission will be slower. Okay, clear? So only the myelinated neuron transmits the signal in a faster mechanism. So now we will look into the basis how the current flow is taking place. So before that, you should know few terminologies to understand this concept. The first one is when you take a cell, whether it is a nerve cell or a muscle cell or any other type of cell. So the cell, when it is remaining in the normal resting stage, you call it as polarized state. Okay, it means the cell is unexcited. Okay, during that period, when you take an unexcited cell, the cell with inside charge is negative. The outside of the cell will be positive. So this is the condition of a normal resting cell or the polarized cell in the unexcited stage. And when the cell is excited, you call it as depolarized. That is excited. Okay, so in the sense there is some change which is taking place with this charges. What is those changes we are going to see? So just now remember, depolarized, it means it is deviated from the resting stage. So that is depolarized. And now coming to the repolarized. Okay, as the term repolarized, it means returning back to the polarized. D is deviated. This is returning. So once when there is a presence of stimulus, okay, so the stimulus is required to make the cell move from resting state to the excited state. Okay, so the stimulus is required for the for making the cell to move from resting polarized state to the excited state. Okay, the resting polarized state is minus 70 millivolt because we have seen that polarized is always negative. So negative charge is more inside the cell during the resting state. So when there is a presence of stimulus, so it is initiating, okay, when there is a presence of stimulus, it is initiating the signal. It is initiating the signal to make the cell to undergo depolarized. Okay, so depolarized in the sense deviating from the resting state, making it to get excited. So for that process, for making the cell to become excited, okay, so what is required for the excitation? The cell which is negative inside during the resting state, it has to become positive. So normally the cell is negative in the resting state. So in the presence of a stimulus, okay, so you are making the opening of the sodium channels. So once when you open the sodium channels, what will happen? The cell will make the entry of sodium ions. So the cell will become positive. Up to the level of 0, okay, from minus 70 up to the level of 0 level, slowly there will be opening of the sodium channels. Slow entry will be there. Okay, and once when it reaches the level of 0, okay, so it makes the faster excitation process, that is called as overshoot process. After the 0 level, overshoot mechanism will take place. So now it gets the help from the potassium ion also, which is another positive ion. Opening of the potassium channel, this is electrical gradient dependent. Because of this positivity inside by the sodium ion, it gets the help from the potassium ion also. Potassium now will get the signal from the electrical gradient. The opening of the potassium ion occurs and the potassium ion also enters. So there will be faster excitation process. So how long? Once when it reaches the peak. Okay, once when it reaches the peak potential is reached. Up to that, the potassium channels will remain open and allow the entry of potassium ion. So now positivity is brought inside first by the slow sodium channels, followed by the overshoot of zero level, and the potassium channels has also participated and it has reached the peak. It has reached the peak in the sense the cell has become excited. Okay, so we don't want over excitation of the cell. So now what happens? The threshold stimulus has done its job. It has made the cell to get excited, but it will not allow the over excitation. So what will happen next immediately is the sodium channels will close. Okay, once when the sodium channel is closed, next thing immediately is the entry of positive ions inside will be interrupted. Once when the sodium channel is closed, okay, the sodium channel is closed, so no more positive ion is entering inside the cell. Okay, and also together what help is being given by the potassium channels? It makes the potassium ions to enter inside. But the job is done. Peak potential is reached. Excitation is reached. So the potassium ions leaves the cell. So the positivity will come out of the cell and makes the cell to become negative slowly. So no sodium entry, no potassium entry. Okay, no sodium entry, no potassium entry, and along with that there is exit of potassium. Okay, so this makes negativity to come back as like that of the resting state. So that is called as repolarization. And here you called as depolarization. Okay, so during this repolarization process, finally it has reached the resting level. But this is the resting level. During the exit of the positive ions, it will also remove more and more of positive ions. Okay, so that makes more negativity, and that part is called as hyperpolarized, but that is only for a short time. Then it will restore back to the resting level. Okay, so this is what is called as action potential. Remember, where is the opening of sodium channels, where is the opening of potassium channels, and where is the closure of sodium channels, where is the closure of potassium channels. Okay, so this is what you have to remember. Okay, so depolarized, repolarized, hyperpolarized, and the excited state. The next we are going to see is the different types of nerve fibers. See, this types of nerve fibers is very important. So now going on to the types of nerve fibers. This types of nerve fibers and the classification is very important, because in this also there is a question in this past December 2021 exam, okay, asking that a clinical scenario given, followed by which nerve fiber is having the faster conduction and which nerve fiber is having the receptor for pain sensation. So we cannot determine only this is important, only that is important. Overall, this classification of nerve fibers is important. Okay, so try to remember this picture because this picture will give you a much information with all the aspects regarding the diameter, regarding the velocity, regarding the faster conduction, regarding the myelination, everything. Okay, so try to at least remember this picture in your mind, so the visual memory will help you if you are forget during the exam. Okay, so this classification of nerve fibers is given by, so Erlangers and Gasser is the person who has given this classification of nerve fibers. So this Erlanger and Gasser has classified based upon the fibers as A fibers, B fibers and C fibers. And there is also another classification of nerve fibers, that is numerical classification of nerve fibers, where they have given type 1, type 2, type 3, type 4 in the terminologies. Okay, so altogether both are determining the same. Okay, so first we will see the Erlanger and Gasser's classification. They have classified type A, type B and type C fibers. This type A fibers is further classified into A alpha, A beta, gamma and delta. So this A alpha fibers is recognized for somatic motor neurons and for the proprioception. So A alpha fibers is meant for somatic motor neuron and for proprioception. Proprioception in the sense it is for positional sensation. So it is the receptor which is present in the muscle spindle, especially the skeletal muscles. Okay, and also in the Golgi tendon organ. So skeletal system mainly have the skeletal muscles mainly have the muscle spindle and the Golgi tendon organ, which is required for positional sensation. Okay, so that is A alpha fibers. And then next is the A beta fibers, which is meant for touch and pressure. Okay, the touch and pressure sensation is by the A beta fibers, and followed by the gamma is motor response to the muscle spindle, whereas alpha is sensory to the muscle spindle, whereas gamma motor neuron. Always we will tell and we will study in most of the places gamma motor neuron, gamma motor neuron. It means gamma is for motor. Remember that. Whereas A alpha is also for muscle spindle, that is sensory. And delta fibers. Remember, this is for pain and cold, also for touch. And pain here it is categorized only for the fast pain. Delta fibers is fast pain sensation. And you have C fibers, which is also for pain, that is slow pain. Remember, C, slow. Spell the slow as CLOW. So that is C fibers. This is just for your understanding and remembrance. Okay. Fast fibers, delta fibers. Okay. And slow fibers is C fibers. So in pain sensation, fast pain and slow pain is mediated by two different types of nerve fibers. Fast pain, slow pain. This based on this pain sensation only, a question was asked in this past December 2021 exam. Okay, clear? So remember this. This is the twisty part. That is the reason why they have asked the question in this. And another twisty part in this classification is muscle spindle. Sensory is from A alpha and gamma is from motor in the muscle spindle. Okay, remember. So fast pain, delta fibers, slow pain, C fibers. Okay. And type B fibers is sympathetic preganglionic, whereas type C is sympathetic postganglionic. B comes first, then C. Okay, so this is preganglionic, this is postganglionics. This is another just way for remembrance without any confusion. So preganglionic, postganglionic, both for sympathetic. Okay, B fibers, preganglionic sympathetic, C fibers, postganglionic sympathetic. A, B, C. A after B, then C. So B is first, so preganglionic. C is next, postganglionic. Just the way for remembrance without any confusion. Okay, clear? So, and then followed by the C fibers, the dorsal root fibers is carrying the pain receptors, hot sensation, and other mechanoreceptors. Okay, so these are the most important part where you will feel the twisty questions. Okay, with respect to the pain fibers, fast pain, slow pain, and sympathetic preganglionic, sympathetic postganglionic. Among A fibers, B fibers, and C fibers, which is most numerous is the C fibers. Okay, clear? And which is myelinated fibers, that is also given in the green line representation here, is A and B fibers. Myelinated fibers, C is non-myelinated, but that is most numerous, and that is the reason why it is carrying the slow pain sensation. Always myelinated fibers will perform faster conduction, whereas unmyelinated will perform slower conduction. So slow pain, C fibers, it is unmyelinated. Just integrate your functions and remember. Okay, myelination, faster conduction. So B fiber is fast pain. C fiber, unmyelinated, slow conduction. Slow pain is C fibers. But unmyelinated fiber is most numerous. Clear? Clear with this? And follow this red arrow in the left hand corner. Okay, so it is mentioning the diameter and the velocity. It is decreasing, descending. It means that A fibers is increase in the thickness, increase in the velocity, which is less than the A is B, less than the B is C. So the order is decreasing. Okay. Okay. So now this Erlanger and Gasser classification is also being termed numerically. Okay. So that is A alpha fibers, which is giving rise to the muscle spindle, that is being termed as 1A fibers. Okay. And A alpha fibers to the Golgi tendon organ, that is 1B fibers. Okay. And B fibers for touch and pressure is type 2 fibers. Clear? And then delta, which is a fast pain sensation, is 3 fibers, type 3 fibers. So, and then C fibers, which is slow pain sensation, and for hot sensation, that is type 4 fibers. So numerical classification is type 1, type 2, type 3 and type 4. Type 1 subclassified as type 1A and 1B. Okay, clear? So this is the classification of nerve fibers based on myelination, based on the diameter and velocity, and based on the functions, and based on the numerical classification. We have different case classification patterns in the nerve fibers. And there is one more classification of nerve fibers based on the polarity, in the sense. So, for example, this is a neuron. Okay. So, when the neuron is giving rise to many processes, short processes and one long processes. So, this is a multipolar neuron, which is the neuron which we have in our body. So apart from that, there are also neurons where the cell body, first one, unipolar neuron, the cell body do not have multiple dendrites. Only one processes will come and that will give rise to the dendrites as well as the axons. That is unipolar. And there is bipolar, only one dendrite and one axon. And pseudo-unipolar, where from the same process, axons and dendrites will rise. And the multipolar is most common, which is the normal neuron. Okay, based on the polarity also it is being classified. And this is the diagrammatic explanation of, this is the muscle spindle, which is the receptor for the skeletal muscles. Okay, so this muscle spindle, you have the, now this is the structure of a muscle spindle, where you have this bulged portion you call as nuclear back fibers, and these are all nuclear chain fibers. And type 1A is the sensory for muscle spindle, which supplies type 1A, which give rise to both nuclear back fibers and nuclear chain fibers, whereas type 2 will give only for nuclear chain fibers. Okay, so the gamma motor neuron, which is the motor which is present in all the muscle spindles. Okay, so this is just the diagrammatic representation of the muscle spindle and its nerve fibers distribution. And moving on to the synapse. What is meant by synapse? Synapse, it is the junction between two neurons. When you are seeing the structure of a synapse, this is the axon terminal of one neuron, and this is the receiving neuron. Okay, so this is called as presynaptic, and this is the postsynaptic terminal. Okay, presynaptic is the neuron which is giving the signal, and the receiving neuron is the postsynaptic. So in between these two neurons, you have the junction that is called as synapse. So if you look into this structure classically, the presynaptic neuron and the postsynaptic neuron. So in between these two neurons, you have the space that is called as synaptic cleft. Okay, and this presynaptic neuron will contain numerous vesicles. These vesicles are the one which carries the neurotransmitters, which has to reach the postsynaptic membrane. So these neurotransmitters we have seen in the vesicular transport. The neurotransmitters, how it is transported, the neurotransmitters will come and reach the vesicles will come and reach the presynaptic membrane, and by the process of exocytosis, the neurotransmitters will be released in the synaptic cleft. Okay, so those neurotransmitters which is released in the synaptic cleft, the postsynaptic terminal will have the receptors for all those neurotransmitters. So these neurotransmitters will come and bind with these receptors. Now, remember the action potential here. Once when the receptors attached with the neurotransmitters, that is the stimulus here. Okay, so this stimulus will activate the postsynaptic membrane, resulting in the development of action potential and making it to get excited. The work is, function of the neuron is initiated. Okay, so that is why action potential is the very most important basic when you are studying about the nerve physiology. Not only for the nerve physiology. If in case instead of a postsynaptic neuron, if it is a muscle, you term it as motor end plate. So once when the chemicals reaches the muscle, then the muscle contraction will happen. Okay, muscle contraction will happen in the presence of action potential generation. So that is why this action potential is very, very important concept. So exocytosis, the cell physiology is applied over here. By the process of exocytosis, the vesicle is getting ruptured with the postsynaptic membrane, and now it is reaching the synaptic cleft. Okay, then in the synaptic cleft, it is attached to the specific receptors on the postsynaptic membrane. Then this is considered to be the stimulus, and this stimulus activates the sodium potassium channel. Okay, opening of sodium channels, entry of positive ions, depolarization, then the function is done. Okay, so that is the synapses. And now moving on to the muscles. We have different types of muscles. Okay, so this types of muscles is being classified based upon the striations: skeletal muscles, cardiac muscles, and smooth muscles. When you look into the skeletal muscle and the cardiac muscle, they both are striated muscles, whereas smooth muscle is non-striated muscles. And this skeletal muscle is voluntary. In the sense, if you want to flex your arm, flex your hand, you can flex. If you want to extend, you can extend. But the cardiac muscles and the smooth muscle, involuntary. In the sense, you cannot control its function. You cannot control the function of a heart. I am tired, wait for a few minutes, don't pump. You cannot do that. In same way, for smooth muscles, if you cannot give signal for the kidney to stop functioning, for stop forming the urine for 1 day, you cannot do that because it is involuntary. Only the skeletal muscle control is in your will. So that is voluntary muscles. Okay. So next, moving on to the muscle filaments. So if you take a muscle, the whole muscle, you call it as a muscle belly. And this muscle, when you take a cross section, the outermost covering of the muscle, you call it as epimysium. And this epimysium will contain numerous fascicles. Okay, so each fascicle is covered by perimysium, and this perimysium, which is in the fascicle, it contains multiples of muscle fibers. These multiple muscle fibers contains the myofibrils, and these myofibrils is made up of muscle filaments, that is thick and thin filaments. These thick and thin filaments play a major role in the muscle contraction process. So this is the thick and the thin filaments. So you have, when you go into the muscle filaments, I will just tell you, thick filaments, it is made up of myosin, and thin filament, it is made up of actin, troponin, tropomyosin. Okay, so this arrangement of thick and thin filaments, it forms the striations. These are thick filaments, and the blue colored lines, they are all the thin filaments. So alternatively, thick and thin filaments are arranged. So these arrangement of thick and thin filaments is the one which is giving you the striated appearance in the skeletal muscles and the cardiac muscles. Okay, so the space between this is the Z line. So the space between two Z lines, you call it as sarcomere. So this sarcomere is the functional unit of a muscle. The functional unit of living organism, you call it as a cell. In case of muscle physiology, the muscle cell, you term it as a sarcomere. So this is the single structural functional unit of a muscle. Okay, so the detail of how this muscle physiology, muscle contraction is working on, we will see in the regular classes. And the myosin, this is the structure of the myosin. You have the tail and the head. Okay, so in the head, you in between the tail and the head, you have a flexible hinge region, also the neck part. Okay, so this head portion is the one which is flexible, in the sense it makes the myosin to move from forward and backward for the muscle contraction to perform. So the myosin head will move like this. Then in the next step, it will move like this. Then it will move like this, and it will help in the muscle contraction. And this is the structure of thin filaments. So thin filaments is made up of actin, troponin, and tropomyosin. So the globular structures, a bead-like structures. So a row of beads, arrangement of beads. The globular portion, you call it as actin. Okay. So only just the globular proteins cannot form the stable structure. So in order to maintain the stability, a thread-like structure will also follow the actin, that is called as tropomyosin. Okay, and this actin contains some specific protein or the specific site which you called as myosin binding site. So this troponin, when it is covering, it will cover all the myosin binding site. Okay, only when there is a need for the excitation of a muscle, when the muscle is about to be contracted, in those regions, the myosin binding site will be exposed. In other cases, it will not be exposed. And in order to maintain the tropomyosin and actin stick together, you will also have another structure that is called as troponin, which is present in between. And this troponin has three sites: I site, C site, and T site. I site binds with the actin, C for calcium, T for tropomyosin. So, okay, so this is the structural representation of how the thick filaments and thin filaments are present. So, and in detail of all these functions, how it is doing its function, how the mechanism of muscle contraction is taking place, we will see in the regular physiology classes. And moving on to the another important thing is the neuromuscular junction. As the same concept, synapses between the neuron, and this is the junction between the nerve and the muscle, as the name, junction between the nerve and the muscle. So same mechanism, you have a presynaptic and the postsynaptic membrane, where the postsynaptic membrane, here in this case it is a muscle, so it is called as motor end plate. Okay, so here also the presynaptic membrane will have the vesicles which contains the neurotransmitters, and the vesicles will be present in the axon. So once when there is a stimulus, there will be entry of calcium. In case of muscle neuromuscular transmission, you have the role of calcium. This calcium makes the vesicles to move towards the presynaptic terminal. Then once when it reaches the presynaptic terminal, by the same process of exocytosis, it is released and binds with the receptors in the postsynaptic motor end plate. Okay, and then it will attach to the receptor that is considered to be the stimulus for the motor end plate. Again, here action potential is generated and the muscle will perform its function. The events in the neuromuscular transmission is Very, very, very important. You should know the order. First, what is happening. And the same December 2021 exam. What is the first event which is happening in the neuromuscular transmission is being asked. The answer here is entry of calcium ions. So this is one more exam question which was asked in the December 2021 FMGE exam. So in the neuromuscular transmission, calcium entry makes the vesicles to move towards the membrane and allows the exocytosis process to perform the function. Okay, so this is the important concept which you have to understand. And once when the muscle contraction is happening, you term it as excitation contraction coupling. The detailed explanation of the mechanisms, how it is doing the function, we will see in detail. Okay, so first and foremost here, the entry of calcium plays a major role. So calcium ions in the case of skeletal muscles, it is from the extracellular fluid. Remember this, the calcium source for the skeletal muscle contraction is from the extracellular fluid. Whereas in case of cardiac muscle contraction, the calcium is from both extracellular fluid, also from the sarcoplasmic reticulum. Okay, clear? So this is the important thing in the cell and nerve muscle physiology. So endocrine system, as the name endocrine, it is going to release the endocrine hormones. Endocrine hormones is different from the exocrine chemicals. So endocrine hormones, they are also the chemicals, but it is produced in the glands, specific glands, and those glands will release those hormones, and these hormones will reach the target cells. First and foremost, what are all the endocrine hormone glands we have? Okay, so start from the top. In the brain, we have two major important endocrine hormones, glands, the pineal gland and the pituitary gland. So pineal gland, detail of all those things we will see in the regular nervous system physiology. And some pituitary gland notes I will give you now. Pituitary gland, followed by that in the neck region, we have thyroid gland and the parathyroid gland. Next, adrenal gland, which is also called as suprarenal gland, which is located in the upper portion of the kidneys. Then you have the pancreas, which is considered to be the exocrine and endocrine organ, and the reproductive system, ovary, test. So anterior pituitary hormones. In the anterior pituitary gland, you have the acidophilic and basophilic cells. So you should remember what are all the hormones acidophilic and what are all the hormones as basophilic. Okay. So prolactin and growth hormone are acidophilic, whereas basophilic is FSH, LH, ACTH, and thyroid hormones. So you can remember with the mnemonic FPG got flat. Okay, A stands for acidophilic, B stands for basophilic. The hormones of acidophilic is P and G, prolactin, growth hormone. The hormones of basophilic is FSH, LH, ACTH, and thyroid hormone. Okay, so this is another important exam question which was asked in the previous exam. Okay, acidophilic, basophilic, based upon that. Okay, so the hormones of anterior pituitary is thyroid hormones, ACTH, FSH, LH. You call it as gonadotrophic hormones, growth hormones, prolactin, and endorphin. So these are all the hormones of the anterior pituitary gland. So among the hormones of the anterior pituitary gland, only 2-3 hormones we are going to see over here with some concepts only. Okay. So the first and foremost is the growth hormone. So growth hormone, as the name, it is responsible for the growth of all the cells, tissues, bones, etc. Okay. So mainly these growth hormones is taking part its role in the growth of bones, cartilages, and connective tissues. So overall, it is responsible for the ossification. Okay, so this growth hormone, it has been maintaining the ossification and bone formation process. Whereas in case of metabolic functions, in proteins, it has been responsible for the anabolic metabolism. In fats, this is responsible for the lipolysis. Since it has been involved in the anabolic metabolism of the proteins, this plays a major role in increasing the body mass. In case of fat metabolism, it is performing the lipolytic function. So this is responsible for the ketogenic effect. Whereas in case of carbohydrate metabolism, this acts as a diabetogenic effect. So the growth hormones in proteins, anabolic, in fats, ketogenic, in carbohydrates, diabetogenic, by synthesizing new glucose molecules. Whereas in case of minerals, calcium absorption, sodium retention, all those things. Whereas it is also having its major role in erythropoiesis and the development of mammary glands, lactation, etc. Okay, so these are all the some major functions of the growth hormone. So if you have some defect in the functions of the growth hormone, either it is hypersecretion or hyposecretion, you term it as gigantism and dwarfism. So we have seen this is playing a major role in the ossification of the bones. So for example, if you take a bone, okay, you have epiphysis, diaphysis. Okay, so diaphysis, epiphysis. During the bone formation, normally there will be the fusion of epiphysis with the diaphysis. So if there is excess of growth hormone before the fusion of epiphysis with the diaphysis, it results in the elongated bone. So instead of this normal bone structure, you have the elongated bone structure. So this increase the length of the bones. Okay, this is happening before the fusion of epiphysis with the diaphysis. So that results in the feature called as gigantism. Okay, clear? So before the fusion of epiphysis with the diaphysis, excess of growth hormone is present in the body. So it makes excessive lengthening of the bones and resulting in a condition called as gigantism. So this is the normal person. And if you look into the gigantic person, he will be very, very, very tall. Lengthy bones, the long bones, femur will be very long, and then you have the humerus is long, everything is long. So the person will become gigantic. And next one is the dwarfism. Dwarfism is the condition when there is a deficiency of growth hormone before the fusion of epiphysis with the diaphysis. So instead of this normal length of the bone length will be very short. And there is no growth hormone, so the fusion is also occurred. So after the fusion, there is no chance for increasing in the length of the bone, so the person will become dwarf. But in case of dwarfism, the growth hormone is responsible for only shortening the length of the bones. But the person, normal IQ, puberty will be normal, other things will be normal. Mental intelligence, everything will be normal. Okay, So that is gigantism and dwarfism. This both gigantism and dwarfism is seen in case of children. That is before the fusion of epiphysis and the diaphysis in respect to the growth hormone level. But if the person is encountering a problem in adults, that you call it as acromegaly. Okay, so acromegaly is the condition where there is a defective growth hormone, excess of growth hormone after the fusion of epiphysis with the diaphysis. So what will happen here is epiphysis has fused with the diaphysis. So the person cannot increase the length of the bone, but instead the thickness of the bones will be increased. Okay, so the bone thickness is increased. So that is the condition called as acromegaly. And this growth hormone is also having its role in protein metabolism, fats, carbohydrates, minerals, development of mammary glands, and then lactation, and also for erythropoietin synthesis. For all the metabolic functions, growth hormone plays a major role. If growth hormone is excess in case of adults, whatever metabolic function it is mediating, everything will be in excess. So first and foremost, the bone will be very thick. So resulting in the very increased body mass. Okay, and followed by hypertrophy of the sweat glands. And since it is having its role in the mammary glands, so mammary gland development will be very large, and also it is responsible for lactation. So the person will encounter galactorrhea. Galactorrhea is not common with the females if they are not lactating, and it is also not common with the males. But if a male is encountering a growth hormone excess, acromegaly, they will encounter galactorrhea. And cardiomegaly, because it is involved in the metabolic functioning. Excess of metabolic functioning leads to cardiomegaly, sexual dysfunction, and also peripheral neuropathy. Okay, visual field defects. Why visual field defects is because you have seen the pituitary gland is located in the brain at the portion of sella turcica. So that is the region where the optic chiasma is crossing at the region of the pituitary gland to the occipital lobe. So if the person is having acromegaly, it means it may be due to what is the cause for this excessive growth hormone in adults. You have to look into that. Most probable cause will be pituitary adenoma. So the enlargement of the pituitary gland will be seen. So this enlargement of the pituitary gland will compress the optic chiasma, leading to the visual defects. Okay, most commonly the visual defect due to the optic chiasma is bitemporal hemianopia, and all the bones will be increased, so prognathism will be seen. That is, enlarged jaw will be seen. Okay, abnormal glucose tolerance, because it is also having its role in this glucose metabolism, and orthosis, osteoporosis, because of excess of calcium absorption. Okay, so these are all the common features we will encounter in case of acromegaly. And hyperprolactinemia, this is another anterior pituitary hormone defect. So hyperprolactinemia might also be due to the pituitary tumors. So excess of prolactin level. So finally, prolactin is increased. If the prolactin is increased, means normally prolactin is increased during the lactation. Instead, this lactation increased prolactin will result in amenorrhea. But if the person is not lactating, so in the anterior pituitary, we are now looking into the prolactin hypersecretion. Okay, so you just continue with the class. Since we are running out of time, I was not able to repeat it as like action potential. Clear? This is hyperprolactinemia. Normally, prolactin level will be in excess after the parturition process. Once when the estrogen and progesterone is decreased after the delivery, once when the delivery is done. So that makes the prolactin to be in excess. Prolactin is a lactating hormone. So this lactation will also makes the prolactin to inhibits the growth gonadotrophic hormones. Okay. So this leads to amenorrhea. This is normal in the lactating women. But in case of hyperprolactinemia without lactating women, okay, so that is not considered to be the normal. So the cause will be most probably anterior pituitary tumors. So tumors of the anterior pituitary, the most common one is hyperprolactinemia. Prolactinoma is the most common one. So this prolactin excess, it will cause decrease of gonadotrophic anovulation, and this results in infertility. So you have to look into that. If a person is coming with amenorrhea without any history of lactation, then you have to check with the prolactin levels. And there will be also secretion of milk, that is galactorrhea, without any history of lactation. So that is the most common thing you have to encounter. Normally, prolactin level will be controlled by the dopamine. So dopamine acts as a control, inhibitory role in the prolactin secretion. But in case if there is a decrease of dopamine, when the person is encountering or when the person is already having a history of any intake of antipsychotics, so that will decrease the dopamine level, and followed by the antipsychotic drugs, you will also encounter this hyperprolactinemia. So hyperprolactinemia, prolactin is not just the lactotrophic hormone. It is also having major role in maintaining the fetal functions. And if a person is given with antipsychotics, followed by that, if they are encountering hyperprolactinemia, we have to look into that. So this is a short gist about the anterior pituitary hormones. And the other anterior pituitary hormones or thyroid hormones, then ACTH and other things we will see separately in all the individual glands. So coming to the posterior pituitary gland, we don't have much hormones like anterior pituitary gland in the posterior pituitary gland. So posterior pituitary gland have only two hormones, that is ADH and oxytocin. This is controlled by the neural mechanism. So neurohypophysis, all the hormones, both ADH and oxytocin, synthesized by the supraoptic and paraventricular nucleus of the hypothalamus, and through the neural signaling, it is stored in the posterior pituitary gland. Okay, remember, posterior pituitary hormone synthesized in the hypothalamus, not in the posterior pituitary gland. Synthesized in the hypothalamus, stored in the posterior pituitary gland. It is just releasing when there is a need for the hormones. Okay, so this ADH is having its main role in two mechanisms. One is, it is also called as vasopressor. ADH, it is also called as vasopressor, in the sense it is having its role in the contraction of the blood vessels. So that is required for the maintenance of blood pressure. And the other one is, as the name ADH, antidiuretic hormone. So antidiuretic hormone is, it is having its role in the control of water retention during the urine formation. So this is the only two function of the antidiuretic hormone. One is a vasopressor, which is a major role in the blood pressure regulation, and the antidiuretic hormone, which is having its major role in the water balance. Okay. So if there is abnormality, if you are encountering abnormality with the antidiuretic hormone, you will see the two common clinical features as SIADH, syndrome of inappropriate antidiuretic hormone, and diabetes insipidus. So normally the ADH, this is with the abnormality of ADH. So we have seen ADH. This is the second function of the ADH, maintenance of water level. Okay. So this ADH is having its role in the distal convoluted tubule and collecting duct of the kidney. So what this ADH will do is, I will be quite fast, but please cooperate. I will just tell you the common functions. Clear? So this ADH will act on the aquaporins in the nephron, collecting duct and the distal convoluted tubule, okay, to retain the water. Okay, if there is decrease of ADH, that leads to excess of water release. You term it as polyuria, which is a common clinical feature of a diabetes mellitus. But here it is also called as diabetes, but it is insipidus. This diabetes insipidus is due to the deficiency of ADH. So when there is a deficiency of ADH, it will not have the role on the aquaporins. It will not activate the aquaporins. Aquaporins are the proteins which is getting activated only when the ADH binds to it in the nephron. Once when it binds to the nephron, then only it will do the retention of water and control the urine volume. If there is no ADH, no aquaporin activation, no retention of water. In the sense, excess of water will be released from the kidney nephron and leads to polyuria. So the person will encounter water loss. That is, the person is dry inside. Only the water is being released in the form of urine. That is diabetes insipidus. Whereas on the other side, soaked inside. See the reverse of this diabetes insipidus. The person is soaked inside. It means there is excess of ADH. It is also due to the pituitary tumor. Okay, the cause may be of anything, but most commonly pituitary tumors increases all the hormonal level. So what will happen? More and more of ADH action on the aquaporins, more and more of water retention. So the person will be edematous. There is, here you have encountered polyuria, but here it is oliguria. Only very less volume or no urine anuria. Okay, so that is, you call it as a syndrome of inappropriate antidiuretic hormone. So this is the two common clinical features you will encounter in the ADH. And the another one is oxytocin. One of the function of oxytocin is in the process of parturition. That I have explained you when I am explaining the homeostasis. And the another mechanism in the homeostasis, positive feedback mechanism, during the delivery process, excess of oxytocin is being produced. So it will make the uterine muscles to relax and favor the delivery of the baby. So that is one of the function of oxytocin. And the another function of oxytocin is in the process of milk ejection. See, again another twisty concept over here. Oxytocin is a milk ejecting hormone, but milk secretion, it is by the prolactin. Don't get confused. Milk secretion, production is by prolactin. Whatever milk which is produced by the prolactin in the mammary gland, it is stored in the mammary gland. but it will be ejected, released by the oxytocin. Okay. So once when the baby sucks the nipple of the mother, that will give you the sucking reflex to the hypothalamus. Then the hypothalamus will produce more and more and more of the oxytocin and give it to the posterior pituitary gland. This posterior pituitary gland will come and give signal to the mammary gland to eject the milk. So that is the another function of the oxytocin. Okay. So that is about the pituitary glands. And next, followed by the pituitary gland, you have the thyroid gland. Thyroid gland, which is present in the neck region anteriorly. Okay. It is a butterfly shaped organ. Okay. This picture was asked in December 2021 exam. They have labeled this and asked what is the hormone synthesized by this structure. So this is the histology picture, but it is also asked in the exam. So you should know the pituitary gland contains the follicular cells. Inside you have the colloid. So this colloid is the place which was asked in the exam where the secretion, production of T3, T4 is taking place. So this is one follicle, this is one follicle, this is one follicle. And you also have the parafollicular cells. So this is the follicular cell colloid. So they are involved in T3, T4 secretion. The option is like thyroxine, I think. So this is exam question. Okay. And they have asked what is the hormone produced. So at the same time, you should also know about the parafollicular cells of the pituitary gland. So the parafollicular cells, also called as C cells, which produces another hormone called as calcitonin, which has a role in the calcium homeostasis. So don't think that this is not important in any of the concepts, not only in physiology I am telling. So all the basics you should know. So mostly when the people is looking into this picture, they will just look, oh, this is the thyroid gland. No, only the follicular cells that is releasing the thyroid gland, thyroid hormones. That's it. Don't think like that. If you are looking into the concept, understand the concept clearly and completely. So that is the most important thing you should learn in this past exam question. December 2021 exam is full and full of basics. Okay. Even in the OB-GYN cases also, they have given a case, but the question was framed like, what is the epithelial cell of the fallopian tube? So, but they have given a very big case for that. And then finally, the epithelial cell of the fallopian tube is the key question in that question. So don't think that the clinical subjects is important, preclinical is not important. Don't always judge like that. Whatever concept you are studying, study it completely, clearly, understand the concept. Remembering the concept is just made by understanding. Don't mug up the concepts. Clear? So that is very, very important. So now moving on to the thyroid synthesis. So thyroid synthesis, again the same picture, the follicular cells containing the colloid. Okay. So what will happen over here from the extracellular fluid? The colloid will get the iodine molecules. So that will go into the colloid, and in the colloid, it will bind with the tyrosine with the help of thyroglobulin, and it forms monoiodothyronine. Okay. So this monoiodothyronine with another monoiodothyronine, it forms diiodothyronine. Then if it binds with diiodothyronine, it will give T3 DIT plus another DIT, it will give T4. Okay. So T4, you will call it as thyroxine. T3, just triiodothyronine. Okay. So this T3 and T4 enter into the circulation for target functioning. The most important function is it increases the metabolic activity. So as like the growth hormone, thyroid hormone is also having its role all over the body. Okay. Greatest metabolic activity is being produced by this thyroid hormones T3 and T4. Okay, clear? So coming to few regulations of thyroid hormones. So this is the regulation of thyroid hormone. We have seen this in the example of negative feedback mechanism. So from the hypothalamus, you will get thyrotrophin releasing hormone to the pituitary gland that will release TSH, and TSH will release T3 and T4. If T3, T4 is in excess, it will give inhibitory signal to the hypothalamus and pituitary gland and it will rectify it. The same way it is being regulated. And the metabolic activity, this thyroid hormone will go and act on the nuclear receptor, thyroid receptor. T3 will act on the nuclear receptor and it increases the transcription of mRNA. Since it is acting on the nuclear receptors, nuclear level, it is having the greatest metabolic activity. So it is having major role in the metabolic activity. Okay, clear? So some thyroid disorders, if you encounter hypothyroidism, here also you call it as cretinism. Okay, so it will affect the growth, everything, and this is also called as thyroid dwarfism. Remember the difference. In case of growth hormone dwarfism, only the person will have short stature. But in case of thyroid dwarfism, the person will have low mental intelligence, decrease of pubertal functions, and decrease in the metabolic functions, everything. But all those will remain normal in case of growth hormone dwarfism. Okay. If the person is encountering the same hypothyroidism in case of adults, so they have myxedema. So myxedema, main thing is periorbital edema will be seen. And because of the excessive accumulation of hyaluronic acid, the face will be blotted, decrease in the BMR, and they will have cold intolerance. And mental activity is decreased. Sexual functions will be decreased. Amenorrhea leads to infertility and pretibial edema. Also, all these are most common with the hypothyroid functions. And in case of hyperthyroidism, so what will you encounter is mainly there will be exophthalmic goiter. So exophthalmos will be seen. So they will look like the person present, seen in the graveyard. So you call it as Graves' disease. And in Graves' disease, there will be enlargement of the thyroid gland. But most probably, the person will remain symptomless. If this is remaining for a long period of time, because of the Graves' disease, it leads to thyrotoxicosis, where you will produce all the symptoms. Whereas in Graves' disease, mostly it will be symptomless. Okay, clear? So this is a few small introduction about the thyroid disorders. So other thyroid disorders, apart from this hypothyroidism and hyperthyroidism, you have Hashimoto's thyroiditis. This is mostly autoimmune. And also you have Dequarian thyroiditis. So this is mostly by the viral infections, leads to painful enlargement of the gland. Whereas in Graves' disease, symptomless enlargement, but here followed by the viral infection, mainly the giant cell infiltration. So this keywords is the one which you have to remember. The painless enlargement is you can encounter in Graves' disease, mostly painless, symptomless. But the opposite, painful enlargement with the symptoms, is Dequarian thyroiditis. Okay, so these are some of the other thyroid gland disorders. And followed by that, you have the parathyroid gland. So you have two pair of parathyroid glands which is located in the posterior aspect of the thyroid gland. Okay, so this parathyroid gland mainly releases the hormone called as parathyroid hormone from the chief cells of the parathyroid gland. So main function of this parathyroid gland is to maintain the serum calcium level. So this parathyroid hormone mainly increases the serum calcium level. Okay, so how it will increase the serum calcium level is, for example, this serum calcium level is being increased by acting on the bones, by acting on the kidneys, and by acting on the GIT. So this three regions is the place where it will encounter. So in case of bones, it causes demineralization. Whereas in case of kidney, it will increases the calcium reabsorption. In GIT also, it increases the small intestine absorption of calcium. So altogether, it increases the serum calcium level. So this is the function of the parathyroid hormone. So it is having its role over the bones, kidneys, and GIT. In the kidney, it is also having another role, is activation of vitamin D into its active form. That is 1,25 dihydroxycholecalciferol. So that is the active form of vitamin D. So this 1,25 dihydroxycholecalciferol, it is the active form of vitamin D. This is exam question in this December 2021 exam. Okay, the active form of vitamin D, hydroxycholecalciferol, dihydroxycholecalciferol, calcitriol, 1,25 dihydroxycholecalciferol. That is the active form of vitamin D. So whatever concepts you are studying, just understand it completely and remember it completely. So that is the most important thing you have to know. So in case of hyperparathyroidism, whatever function you have seen over here, that will remain in excess. Hyperparathyroidism due to the adenoma of the parathyroid gland. So parathyroid gland will be enlarged. So leads to increased production of the parathyroid hormone. So more action of parathyroid hormone on the bones, causing demineralization. On the kidney, excess calcium absorption, and on the intestine, excessive calcium absorption in the small intestine. On the whole, it has reached its goal by increasing the calcium level, increasing the serum calcium level, but altogether it will also end with few complications like, so hyperparathyroidism So its goal is increasing serum calcium level. But in the bones, excess of calcium will lead, demineralization will lead to osteoporosis. Okay. In the soft tissues, there will be metastatic calcification. Then in the kidney, you have calciuria and you will encounter calcium stones. Renal calculi will be seen. Altogether, small intestinal absorption, nausea, vomiting, constipation. So these are all the complications you will encounter in case of parathyroid hormones. Okay. And followed with the pituitary, thyroid, parathyroid glands, there is another important pathological scenario. You call it as men's syndrome, multiple endocrine neoplasia. So I will just give you a small intro of how to remember. This men is of three types: type 1, type 2A and type 2B. Okay. So remember with this mnemonics, type 1 is triple P, type 2 is TAP, type 3 is TAN. Okay. So this TAP, P stands for pituitary, pancreas, parathyroid. So pituitary, pancreas, parathyroid glands. Whereas TAP stands for thyroid, adrenal and parathyroid. In case of type 2B, this TAN stands for neuroma, but this other two is the same. Okay, so that is about the men syndrome. And then moving on to the adrenal glands, you also call it as a suprarenal gland. When you take a cross section of the adrenal gland, you have the adrenal cortex and the adrenal medulla. Adrenal cortex further has zona glomerulosa, fasciculata and reticularis. So these three structures of cortex and the adrenal medulla, even though they are organized in a small single adrenal gland, they have different functions. Okay. Zona glomerulosa produces mineralocorticoids. Aldosterone is the hormone. And fasciculata produces glucocorticoids, that is cortisol. And reticularis produces androgens, that is dihydroxyepiandrosterone. And medulla produces epinephrine. Okay, so these are all the hormones produced by the adrenal gland. And the regulation of adrenal gland, same from the hypothalamus, anterior pituitary, adrenal cortex. So corticotrophin releasing hormone act on the anterior pituitary, releases ACTH, and this acts on the adrenal cortex to release the cortisol. If you encounter excess of cortisol, then by the negative feedback mechanism, it will be controlled. Okay, the function of glucocorticoids, same like that of growth hormone, enhances the glucose level, increases the new glucose synthesis from the proteins, lipolysis, and increases the muscle efficiency. And it also has immunosuppressive functions and anti-inflammatory functions. Okay, it also acts on the bones, leading to osteoporosis. So if a person is having excess of glucocorticoids, then they will encounter Cushing syndrome. So excess of glucocorticoids leads to moon phase because of the increase of lipolysis. Okay, and all the fat will be accumulated in the face and as well as in the back region, leads to buffalo hump or the fat pads. And increase of protein lysis leads to thin skin appearance. And there will be easy bruisability and ecchymosis, and it enhances the metabolic activity. So cardiac function will be very high, leads to hypertension. And then the person will have thin arms and legs because of the lipolysis, all the fat will be released and the protein will be utilized for the new molecules of glucose synthesis. And poor wound healing because it decreases the white blood cells and the immune function cells. And this glucocorticoids also leads to osteoporosis because it removes the storage form of calcium from the bones, demineralization. And moving on to the mineralocorticoids, the most important is the aldosterone. So aldosterone main function is in the RAS mechanism, renin-angiotensin-aldosterone system. So it works by maintaining the sodium level in the body, retention of water. So aldosterone work together with the antidiuretic hormone and together with the angiotensin 2 causes vasoconstriction. And in the kidney, it plays a major role in the maintenance of reabsorption of sodium and water. So altogether, this renin-angiotensin-aldosterone mechanism is having its role in the long-term regulation of blood pressure. Okay, clear? And now, moving on to the adrenal medulla. Adrenal medulla produces the hormone called as epinephrine. When there is a tumor of the adrenal medulla, it leads to excessive production of epinephrine, and that is called as pheochromocytoma. So pheochromocytoma is the condition mainly because of the tumor of the adrenal medulla. The most common clinical feature is PHE, pheochromocytoma, palpitation, headache, and episodic sweating. Along with this, you will have 5 P's, is the same: palpitation, pain, perspiration, and increase of blood pressure and pallor. So if you encounter all these things in a person altogether, suspect for adrenal medulla tumors. Okay. At last, moving on to the adrenal insufficiency. That is with respect to the ACTH. So adrenal insufficiency is of primary, secondary, and tertiary. So always in the endocrine system, you have the hypothalamus, pituitary, and the target gland. Okay. So here also hypothalamus, pituitary, and the adrenal gland. So where the target hormone is produced, that is considered to be the primary. And all the hormones mostly get signals from the pituitary, that is secondary. And tertiary most, hypothalamus is the head ganglion controlling center for autonomic nervous system, also for the endocrine system. That is hypothalamus. So here in this case, adrenal insufficiency means there is decrease of ACTH. Okay, the adrenal gland mainly is not producing the required cortisol. The cause, where is the cause? Whether it is in the hypothalamus or pituitary or adrenal gland, that will determine primary, secondary, or tertiary. So final thing is decrease of steroids. Okay, in case of primary, if the cause is adrenal, means excess CRH will be normal, ACTH will be normal, but this ACTH acting on the adrenal medulla, that is the place which is defective. So steroid is decreased. Then what will happen to all these ACTH which is in excess? This will get deviated and it will act on the melanocortin receptor, melanocortin 1 receptor on the skin, leading to hyperpigmentation. This is encountered only in the primary adrenal insufficiency. Whereas in case of secondary adrenal insufficiency, hypothalamus signal is normal. ACTH is decreased, means the pituitary gland is not functioning properly to give enough of ACTH. So that is the reason why adrenal medulla is not producing the steroids. Okay, that is secondary adrenal insufficiency. And the third one is tertiary. The problem is with the hypothalamus itself. So CRH is decreased. There is low signal from the hypothalamus, less production of ACTH, automatically decrease of adrenal hormones. Okay, so that is adrenal insufficiency. And moving on to the last endocrine organ, pancreas. It is both exocrine and endocrine in its function. Okay, and this pancreas has three different types of cells. One is alpha cells, beta cells, and the delta cells. Okay, so alpha cells, beta cells, and delta cells. So alpha cells produces glucagon, beta cells produces insulin, and delta cells produces somatostatin. Glucagon is blood glucose increasing hormone, whereas insulin is decreasing the blood glucose. Okay, insulin is the only hormone in our body which decreases the blood glucose level. Other than this, all the other hormones like glucagon, steroids, glucocorticoids, cortisol, growth hormone, thyroid hormone, every other hormone will increases the blood glucose level. So insulin is the only anti-diabetogenic effect, whereas other is having diabetogenic effect. Remember this always. Okay, so when there is a deficiency of insulin, decreased production of insulin, that results in the condition you call it as diabetes mellitus. Okay, so this insulin, what it is doing in maintaining the blood glucose level is this insulin act on the receptors for the insulin in the cells and makes the entry of glucose into the cell, so the blood glucose level is decreased or maintained. But in case of type 1 diabetes mellitus, okay, the pancreas fail to produce the insulin. Okay, so here there is no insulin acting on the receptor, so glucose is remaining in the blood itself, leading to diabetes mellitus type 1. So type 1, there is failure of production of insulin, no insulin at all. Whereas in case of type 2, insulin is there, but the receptor which is responsible for receiving the insulin is not functioning. So this also makes the glucose to accumulate in the blood, leading to excess of blood glucose level, that is type 2. Okay, so know the difference between type 1 diabetes mellitus and type 2 diabetes mellitus. So if you want to tabulate the differences between type 1 and type 2, the mechanism here is beta cell destruction. Cause may be viral infection. There is no beta cell at all in the pancreas. So insulin level is very low or absent. Okay, but here in case insulin is resistant to the receptors. Okay, it means that there is normal level of insulin in the circulation, but the receptor is resistant, so it was not able to act to reduce the blood glucose level by moving the glucose into the cells. But here the genetic predisposition is very weak. Type 1 diabetes mellitus is not hereditary. Okay, that is the meaning for this. But here it is very strong. Okay, type 2 diabetes mellitus is having high predisposition to hereditary. Okay, clear? And the most common complication in case of type 1, the complications like neuropathy, nephropathy, retinopathy, and small vessel atherosclerosis. These are all neuropathy, nephropathy, retinopathy, or small vessel atherosclerosis. If it is happening, the atherosclerosis happening in the large vessels, that leads to stroke, myocardial infarction, all those things. So the complications is very common. But in case of type 2, it is not much common. But if it is uncontrolled, severe, that may also happen. Okay. The most common treatment is you have to give insulin. No other go, because there is no production of insulin at all. But in case of type 2, you can give oral hypoglycemics and insulin if required. Okay. So this is about the diabetes mellitus type 1 and type 2.